fundamentally, when we talk about nervous tissue, certainly we're illustrating the histology of neurons. And we know from high school biology, for example, that neurons basically have three fundamentally distinct parts. The cell body, which is typical of any eukaryotic cell and found elsewhere in tissues, that it's going to have a typical range of cytoplasmic organelles, nucleus, and so on and so forth of a protein-producing cell. The thing that distinguishes neurons from other cell types are its long membrane-bound cytoplasmic processes specialized for conduction of impulses. And certainly a typical neuron is going to have an array of dendrites, and typically a neuron is going to have a single axon. The dendrites are the receptive portion of a neuron, and in a way, the form of a dendritic arbor of a neuron is kind of an indication of what its functional capabilities might be. And as we've indicated, the job of the single axon is to convey action potentials, electrical impulses away from the cell body toward either a target structure or another neuron. Let's look at the cell body first. We've indicated that like a typical neuron, it contains an array of membrane and non-membrane bound organelles typical of any eukaryotic cell. It's going to have a prominent nucleus with a prominent intracellular nucleolus, mitochondria, endoplasmic reticulum, and so on and so forth. Note that in a typical protein-producing neuron, the amounts of rough endoplasmic reticulum are going to be particularly abundant, and when present, this rough endoplasmic reticulum is known as Nissl substance. So recognize that as being a distinct feature of the cytoplasm of a neuron cell body, as well as the proximal parts of a dendrites of a neuron cell body as well. There are also many structural elements to a neuron cell body as well as its axon and dendrites in the form of either microtubules and neurofilaments that form its skeleton. Microtubules are particularly important, as we'll see, in axonal transport, a mechanism whereby precursors and actual neurotransmitters themselves are transported from the cell body where they're synthesized down the length of the axon. And in many cases, neuron cell bodies may be pigmented in the form of containing either lipofuscin or melanin pigments. Melanin pigments are particularly distinct in neurons that are producing catecholamine-based neurotransmitters such as dopamine and norepinephrine. Dendrites themselves, as we know, are the neuronal process that are specialized to be highly branched and transmit information to the cell body. Axons are thin and of uniform diameter, extend from the cell body to whatever the target structure is, be it another neuron or some sort of gland, muscle, or what have you. And as we alluded to earlier, since the axon does not contain rough endoplasmic reticulum, any neurotransmitter or precursor of a neurotransmitter that's synthesized in the cell body must be transported down the length of the axon. And this transport process basically comes in two phases and basically utilizes microtubules. So fast axonal transport is the mechanism most commonly used by neurotransmitters and or their precursors, and it has directionality to it. In fact, it's bidirectional. The direction by which neurotransmitters are transported from the cell body down the length of the axon to the axon terminal is known as anterograde axonal transport. This is an energy-requiring process that's powered by kinesin, an ATPase motor molecule. Retrograde axonal transport, which basically is a mechanism whereby axon terminals can take up and recycle substances, membranes, and the like from the extracellular or synaptic cleft. Retrograde transport will take up those substances and transport substances back along the length of the axon toward the cell body, a mechanism that also utilizes microtubules and is powered by an ATPase motor molecule known as dynein. And certainly at the distal end of the axon are synaptic boutons that participate in specialized junctional complexes known as synapses. And in a typical neuron that utilizes a chemical neurotransmitter, the synaptic boutons will contain different arrays of forms of synaptic vesicles, each with their own quanta of neurotransmitter. In our picture on the next page, you can kind of get the appearance of a typical multipolar neuron. This might be characteristic, for example, of a lower motor neuron whose cell body is somewhere inside the central nervous system, such as in the ventral horn of the spinal cord, and whose axon leaves in a root of that spinal or cranial nerve in this case, and basically goes out and innervates skeletal muscle. The reason we can say that is because in this case the axon emanating from the cell body is myelinated by Schwann cells, meaning that it must be, the axon must be in large part found in the peripheral nervous system. You can see just an illustration schematically of a typical neuron of the nucleus, nucleolus. Note that Nissl bodies are illustrated here and consist of prominent arrays of rough endoplasmic reticulum, Golgi for the sorting, packaging, and post-translational glycosylation of neurotransmitters. And you can see that the initial segment of the axon basically is unmyelinated, and immediately distal to that the myelin sheath appears, and as we'll see, there are two different kinds of glial cells that participate in the formation of that myelin. Now, just to review the directionality and the elements involved in the transport process, you can see in this illustration that anterograde axonal transport is mediated by dynein and also is certainly facilitated by microtubules, whereas retrograde axonal transport, basically associated with recycling of neurotransmitter and membranes, is occurring in the opposite direction, moving substances from the axon terminal back to the lysosomal system in the cell body. Retrograde axonal transport, as indicated here, as we've alluded to, is mediated by dynein. Next, let's look at a view of the three different fundamental ways of staining neural tissue to demonstrate it histologically. And they're kind of illustrated on these three panels in the middle of the slide. Two common ways of illustrating neural tissue are shown on either ends of these. One is to use a so-called Nissl stain, which, as we know, will only react with rough endoplasmic reticulum and therefore will only stain basically the cell bodies of neurons and or glial cells, as well as the proximal parts of the dendrites. So in the image on the left, you see basically the appearance of an area, in this case of cerebral cortex, where basically the Nissl stain is capable of demonstrating the difference in a way, the size and particularly the shape of particular populations of neuron cell bodies, and it also shows their degree of layering or lamination. In the middle panel is a way of demonstrating all of the processes as well as the form of a cell body of a neuron, and this is basically a method that stains a very small population or percentage of neurons in a particular section, utilizing a silver method known as the Golgi method. When it works, and nobody fundamentally understands how it works, it demonstrates not only the neuron cell body, but all of its processes, including the origin of the axon. Third, the other common way of staining neural tissue is simply to react it with what's called a myelin stain, which certainly will only stain axons that are myelinated. It's also known as a fiber stain that will simply illustrate just myelinated fibers and no parts of either the cell body or its dendrites. Some examples of this are shown on the next two slides. First one is a section through the spinal cord that's stained with a Nissl stain. And very clearly you can see in the area at B are some very large neurons visible even at this low magnification in what we refer to as the gray matter of the interior of the spinal cord. In particular, these large neurons are lower motor neurons that are characteristic of the ventral horn of the gray matter of the spinal cord. Outside them is an unstained area indicated by letter C. We call it white matter, and as you can see in a particular Nissl stain, it's unstained because basically the area at C completely surrounding the gray matter, it basically contains nothing but bundles of axons. In contrast, using a myelin stain on the next figure, also a section through the spinal cord, we can see that the area at A, where the myelinated axons are found, are very heavily and darkly stained throughout the area and surrounding the spinal cord, whereas the area at B is largely unstained because this was the area of the gray matter that almost exclusively consists of neuron cell bodies, their dendrites, And the only light staining that you see in the area of B are axons, myelinated axons, that are either entering or leaving cell bodies in that area. Next, let's look at a specialized area associated with nervous tissue, and this is the area of axonal specialization at the neuromuscular junction. And this is the specialized junctional array that's found between the distal end of a motor axon of a lower motor neuron and its distinct features where it synapses with a muscle cell. And basically, at this neuromuscular junction, the synaptic cleft is thrown into a series of subneural clefts or folds, and basically this area is where acetylcholine is released from the axon terminal of the motor end plate and basically interacts with nicotinic cholinergic receptors on the postsynaptic membrane. And in this figure we can see that illustrated are some of the details associated with a neuromuscular junction. Specifically, you can see the axon nerve terminal releasing synaptic vesicles containing acetylcholine, and you can see basically where they interact with postsynaptic receptors in the junctional folds, which are nicotinic cholinergic receptors. Also, what's shown on here are a number of the different clinical elements that affect neuromuscular junction or the axons that lead to them. Specifically, they include peripheral myopathies that involve myelin of axons near the neuromuscular junction that are affected in Guillain-Barré syndrome. And also what's illustrated on here are other problems that affect the neuromuscular junction, such as the antibody binding to cholinergic receptors that's a cause of myasthenia gravis, the area where botulism toxin binds to the presynaptic membrane, the site where curare basically binds to the cholinergic receptors themselves, as well as the antibodies that are raised against presynaptic calcium channels that is a cause of the Lambert-Eaton syndrome. And the details of these deficits are summarized kind of on the next couple of slides. Fundamentally, when we deal with disorders of neuromuscular transmission, there are a variety of disease states and causes of them, including botulism, myasthenia gravis, curare, snake venom, and Lambert-Eaton syndrome, and some of them are summarized clinically in the next couple of slides. Botulism toxin basically prevents the release of acetylcholine by binding to the presynaptic membrane, and basically these patients have weakness initially in muscles innervated by cranial nerves. Then finally, they're also going to have autonomic signs and symptoms, including dry mouth, abdominal cramping, vomiting, and diarrhea due to basically a problem at the location of junctional specializations associated with smooth muscle. Next, in patients with MG, myasthenia gravis, typically this is caused by antibodies that bind to the cholinergic receptors, therefore blocking the binding ability of acetylcholine. And classically, these patients have weakness initially in skeletal muscles innervated by cranial nerves. Particularly in most of these patients, they will have ocular weakness exhibited basically by a bilateral ptosis. And then fundamentally, most of these patients have an underlying thymoma or thymic hyperplasia. And overall, sustained contractions of the muscles affected in these patients typically leads to increased weakness. And patients typically present more likely with the signs and symptoms as they move along in their day and basically get tired. Curare and alpha-bungarotoxin have the same basic effects. They also bind to the cholinergic receptors and block neuromuscular transmission. In essence, having effects similar to those in patients with myasthenia gravis. Patients with LEMS, basically an underlying neoplasm is typically present in these patients. In this case, however, antibodies are raised against the presynaptic calcium channels that disrupt neuromuscular transmission and the release of acetylcholine. Skeletal muscles, again, in the limbs in these patients are primarily affected, as opposed to skeletal muscles innervated by cranial nerves affected in MG. In contrast, however, in patients with LEMS, sustained contractions of affected muscles leads to an increase in strength rather than to a decrease in muscle strength. Next, let's look at the different forms and glial cells that make myelin and the effect of myelin on its appropriate axons. Certainly, axons in the peripheral and central nervous system may or may not be myelinated, depending on, obviously, their functional capability. And fundamentally, when we see or visualize unmyelinated axons, and when you see them in an electron micrograph, for example, they're very distinct in the sense that unmyelinated axons in peripheral nerves are always encompassed and surrounded by the cytoplasm of Schwann cells. Schwann cells, as we know, are the cells, the glial cells in the peripheral nervous system that make peripheral nervous system myelin. In contrast, when we visualize unmyelinated axons in the central nervous system, they are simply what we consider to be bare axons, and they're not surrounded by the cytoplasm of any corresponding glial cell. And we certainly know from physiology the value of a myelinated axon. The myelinated axons certainly are going to be larger in diameter, but more importantly are faster conducting than unmyelinated axons. The two different forms of myelin-producing cells are Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. Very different feature about how much myelin they make, however. A single Schwann cell is only going to be able to myelinate a single segment of a single axon in a peripheral nerve. So just remember that Schwann means single in this case. Oligodendrocytes, which are the myelin-forming cells of the central nervous system, basically are capable of myelinating many segments of many individual axons adjacent to an individual oligodendrocyte. So a single oligodendrocyte is myelinating many segments of many axons, whereas a Schwann cell is only myelinating a single segment of a single peripheral nerve axon. Next, in both the central and the peripheral nervous system, where there are discontinuities between the segments where there is a myelin sheath, what is created is known as a node of Ranvier. Nodes of Ranvier basically are where the membrane of the naked axon is directly exposed to the extracellular space, and this is a site where saltatory conduction takes place because there is a prominent concentration of sodium channels located in the membranes at the area of a node of Ranvier. Again, in a peripheral nerve micrograph, we know this is that of a peripheral nerve because not only do we see the distinct electron-dense appearance of the myelin sheath, but we know this isn't a peripheral nerve because we see in close proximity to two of these myelinated axons the cytoplasm and the prominent nucleus of Schwann cells. And this is a distinct difference from what we would see if we saw a tract of axons inside the central nervous system, where we would be considerably less likely to see any related glial cell associated with them. So now our next comment bears in mind what happens to an axon that's been severed or injured in the peripheral versus the central nervous system. In our examples here, we're indicating that there's been a traumatic site of an injury that in essence has severed an axon somewhere in the peripheral or the central nervous system. So when you cut an axon in the peripheral or central nervous system, basically you get two forms of degeneration. You get what is known as anterograde or Wallerian degeneration, where all parts of the axon distal to the cut completely degenerate because they've been cut off from their site of nutrition, obviously the cell body. All axons basically proximal to the cut nearest the cell body undergo what are known as retrograde degenerative changes, basically that are most evident in the cell body itself, which is going to make an attempt to regenerate the cut axon. Now, the point that we need to make is only axons that are myelinated by Schwann cells will actually have the capacity to regenerate. An axon anywhere that's severed or injured in the central nervous system that's myelinated by oligodendrocytes does not have the capacity to regenerate. So the points that's illustrated here is, regardless of where the axon is cut, the axon will generate sprouts that will attempt to regenerate the axon, but only axons that are myelinated by Schwann cells will actually have the capacity to promote the regenerating axon, because the Schwann cells, particularly the endoneurium of the Schwann cell, basically will remain intact and provide a sheath to guide the regeneration of the cut axon basically guiding it back to its original site where it can reinnervate, for example, in this case, an area of skeletal muscle. So the point is, only axons that are myelinated by Schwann cells have the capacity to regenerate, and in order for the axon in the peripheral nerve to regenerate, the endoneurial sheath, which is in essence a component of the membrane of the Schwann cells, has to be intact to promote and guide the growth of the regenerating axon. And finally, we know that there are two different forms of demyelinating diseases associated with axons whose myelin is formed by either Schwann cells or oligodendrocytes. Oligodendrocytes, when they are affected by a so-called central nervous system demyelinating disorder, the most common of these disorders is known as multiple sclerosis. And basically, multiple sclerosis typically affects myelinated axons in the central nervous system that are found in tracts or fasciculi near the surface of the spinal cord, brainstem, or near the surface of a particular portion of a ventricle. And since this is a central nervous system demyelinating disease, the only nerve that's commonly affected by multiple sclerosis is the optic nerve. Next, let's look at neuroglia, because these cells are what we call the connective tissue or the support cells of the nervous system, because the nervous system in essence lacks connective tissue. There are a wide variety of neuroglial cells. We've already alluded to two types of them. They are oligodendrocytes and Schwann cells respectively that are producing different forms of myelin. But in addition, there are microglia, astrocytes, and ependymal cells that also play a role in different ways as glial cells in the central nervous system. Astrocytes are the most numerous of the glial cells. Typically, they play a role in the overall support of the central nervous system. By their vascular end feet, they contribute to maintenance of the blood-brain barrier, and they also take up excess neurotransmitter and ions in helping to maintain the extracellular microenvironment of nerve cells. Oligodendrocytes, as we've alluded to already, basically their job is to myelinate many individual segments of many different axons exclusively in the central nervous system. They're affected by a central nervous system demyelinating disorder like multiple sclerosis. Microglia are basically the phagocytes of the central nervous system. They're very different in a sense that they're not derived from neuroectoderm, which gives rise to all other glial cells and neurons in the central nervous system. These are cells that are derived from mesoderm that migrate into the central nervous system. They basically are phagocytic cells, part of the mononuclear phagocyte system. These are the cell types that are affected by HIV type 1, and basically HIV does not actually kill nerve cells themselves, but basically it infects the microglia and causes them to produce cytokines that are toxic to neurons. Finally, ependymal cells line the ventricular cavities of the brain and the central canal of the spinal cord, and their job is to maintain the integrity of the ventricular system. And where the ependymal cells are invaginated by choroidal blood vessels, these areas of ependymal cells become modified and form choroid plexus, in particular areas of parts of the ventricles of the CNS that are involved in the production and secretion of cerebrospinal fluid. Finally, Schwann cells are, as we alluded to, the cell types that produce peripheral nervous system myelin. They're affected in peripheral neuropathies like Guillain-Barré syndrome that affects basically myelinated axons in all cranial and spinal nerves, mainly causing motor weakness as opposed to sensory loss.